Yeah, right. Okay, I can even introduce myself. Okay, so hi, I'm Anirvan Sengupta. As you can see, I'm from physics and astronomy in Rutgers University. And can you hear me? Okay, so I was hoping that I would be one of these very relaxed organizers in this meeting, where all the local organizers do the hard work of organizing and the speakers do the hard work of speaking, and I basically just be there and bask in the glory. And unfortunately, okay, we had few of our speakers sort of at last moment decided not to come. And I decided that this is maybe my opportunity to learn better something I'm struggling with. So you are stuck with listening to me about a subject that I'm barely beginning to understand. So 4.5 hours of pure suffering. And just to make it worse, I have to say that in a long time, this is a set of talks which is most probably a little low on the biology end and a little bit more on the technique end. So I see long faces. But then yesterday I was in ICTS and a friend of mine asked me very tactfully, is your talk self-contained? And usually if someone outside biology asks that whether the talk is self-contained or not, that means, do I really have to come to your talk and listen to all these names? Okay, is it not going to be something where in 2 minutes you will pick up a nice math problem for me to think about? And this one would have satisfied that category. So if there are those of you who have been told too many scary things about E. coli and Alzheimer's and cancers and want a quick problem, this is it. So without any further apologies, let's get to kind of the subject topic. I also cooked up, given the time pressures, a name that is very broad and general, model selection for large data sets. I'll actually be talking about something very specific, and you will see what. Okay. So broadly, this falls under the subject, which I will elaborate upon, is that we hear that in biological systems, we have these big data sets. We are in this new age when there are these big files of data. And if your computational expertise ended with Excel, you are a little bit in trouble. Okay? And perhaps in the early days of it, everybody said there's a lot of data, and we should be able to solve everything. And then you start thinking about the complexity of biological system, and you ask, is this enough data to learn anything at all? Okay? And that relates to the question of this problem in biology to some extent, is that it's not like many other very precise subjects like certain areas of physics where the type of model is very highly constrained. There are maybe only few parameters to learn, and you can do some experiments and fix things out, right? So there is this issue of figuring out what the key variables are, right? So that goes under the list of model and variable selection. And then I'll talk about one particular way of going to it, which goes by the name of compressed sensing and lasso, which originated partly in statistics, partly in signal processing. Then I'll talk about how, in some cases, this method manages to solve problems, and then in other parameter regimes it doesn't, and there is some kind of transition in between that. And then I'll try, so I expect to kind of end today here, and tomorrow I hope to try to give you some insight into what the key things are, why is it behaving this way, right? And the last day, I would try to go beyond this particular method of reconstructing signals when there are too many possible variables and really too little data. Okay? Now, I know that this sounds already still very vague. Okay? So here is some kind of obligatory scary picture saying, oh, these days we are getting a lot of data. This is supposed to be some MRI image of a brain. This is some multi-electrode data train. This is some gene expression profile, and this is some genetic similarity thing. I'm just thrown randomly kind of things that many people who want to look at data and try to understand something about biological systems are kind of struggling with. Okay? Now, usually, maybe this was just yeast and only 6,000 genes and only 4 or 5 conditions. This is kind of primitive large data. Now, you have terabytes of data over something.
But on the other hand, when you think about the complexity of biological system, right, you want to understand, depends on what you want to understand, okay? If you are proclaiming that I want to understand all the genetic variation that could cause something, right? Depending on how complex that model is, this much data may or may not be enough. And I'll try to make it even more concrete in a short while, okay? And the second thing that is not often appreciated is the question of how big the space of possible models is. And this is one place where I have learned by sort of experience where kind of biologists excel, right? They have developed the right nose of what not to look at, what to sort of pick up a smell with, meaning it's still a very, very human intuition-driven thing. We can do lots of analysis and produce big lists of potential candidates, but I've seen sort of in real interactions that a biologist often sort of say there is sort of some amount of knowledge that leads you to pick something up and reject something else, right? So actually, we don't have yet a very good way of sort of doing it mechanically. Perhaps we will never have. Now, just so that I am being concrete, so I want to come to something even more concrete. Well, there is sort of the problem in molecular biology where you want to understand how something in the genotype space, this is drawn in a very abstract way, think of sequence variation, okay, map to certain things in phenotype space, and even that is drawn very vaguely. But now you sort of begin to see a lot of biology examples, right? The phenotype space could be certain kind of development in flies, and the genotype space could be mutations you have done to various genes, right? The phenotype space could be our height and weight, right? The genotype space could be very much our genetic variation, maybe measured in terms of SNPs, okay, in the gene, right? So you often want to know what variations correlate with, okay, phenotype changes, okay? And that problem, let me even give you a special example, right? This is from a kind of study that was pioneered by Leonid Krugliak about 10 years ago, right? So the idea is that you want to figure out in gene expression, right? How does genetic variation affect gene expression? Now, of course, you can screw up a gene and there is nothing useful coming out of it. But there are lots of subtle variations where things are working, but the amount of gene product you make changes. Okay? So in the particular experiment, they took 2 strains of yeast, mixed it up, they let them mate, and generate some degree of genetic variation, and measure gene expression from several isolates. Okay? So if you think about what kind of data you get in this problem, okay, you end up measuring expression of about 6,000 genes, albeit in those days with other crude methods like microarrays, but you have 6,000 numbers for each strain, at least up to some kind of scaling. You have genetic variation markers, like single nucleotide polymorphism, where a certain base could have usually 2 different values. Okay? And you can pretty much indicate it by saying, does it take the majority value or does it take the minority value? So a 0 or 1 would tell you what it is. And if you look across the chromosome, there are blocks over which the SNPs states are correlated. So take chunks that are more or less uncorrelated. So if you do that, there are maybe 100,000 places where there are SNPs that are not very correlated with each other. Okay? They can vary pretty much independently. So now I'm trying to forget biology and trying to make it a math problem. You have 6,000 numbers. You have 100,000 ones or zeros, telling you whether it's the baseline thing, 0, or there is a variation there, 1, right? And in their experiment, there are maybe 63 strains, but order 100 strains for which you know the gene expression, you know the genotype, right? And now it's a very standard exercise for geneticists. Here are some quantitative traits, right? Each gene expression is a real number. It's not yes or no, right? You want to predict, okay, or see to what extent you can predict the gene expression phenotype in terms of the genotype, right? So when I said in the beginning that, oh, the genotype-phenotype map sounds very abstract, but here is a sort of kind of a stripped down problem where we are kind of asking a little bit of a reduced version of the question that I'm playing around, I'm kind of expanding around yeast. Okay, it's still yeast, but if I change the genetics a little bit from what looks like an yeast, right? How does the gene expression change systematically, right? And so, okay, sorry, I should hide this. Okay, so here is the math problem. Okay, right? I have a string of zeros and ones. Okay. 100,000 of them, so 100,000 long bit strings of zeros and ones, and I have 6,000 numbers associated with each. Okay, is this enough data to learn how genetic variation affects gene expression? And now I started talking too much, and I realized that I go very fast when I'm talking without any feedback from you, so I'll stop. So some of you hazard a guess. Okay, is this problem soluble, not soluble, something else? If you do not volunteer, I'll pick one of you. So who will be the good guy? Okay, what do you think? Is this I have 6,000 times 100 things to measure, right? Meaning, should I be able to figure out how genes affect things? The data is too less? Well, actually, maybe I have made things unnecessarily difficult. The 6,000 genes is kind of a little bit fakey, okay? Practically every gene is a story. So let's forget the 6,000, right? Take any one of these genes, right? And look at the strain, okay? It has some genetic variation, right? I want to make a predictive model, okay, in terms of those things. What are the things I should be, is it possible, is it not possible, how should I think about it? Okay, give me your first thoughts, random thoughts. And if it is sillier, the better. Yeah, okay. Okay, no, so I have tried to take boxes which are not linked. So otherwise the number is larger. So they are separated enough, and there has been enough mating so that the SNPs are not correlated.
And then you could look at the spatial location. What is known about the spatial location, transcription factors and things like that?
True. So you are saying, okay, I'll look under the lamppost. If this is the gene expression, particular genes to be varied, let us look in the neighborhood.
Of course, you have to look at the whole thing is you have to look at regulatory networks.
That's true. So now you have said some keyword. There are some regulatory networks. So possibly something very far away and even another chromosome can affect it.
Yes.
Right? So you are not scot-free. You cannot say, I look near and left and right of this gene and I'm done.
Yeah.
So again, is it doable? Is it not doable?
So you have to look at prior knowledge, probably, to some extent.
Okay, I am a stupid physicist. I've gotten into this thing. I've gotten a huge file. I know how to code in Python or R. Okay, and I don't want to know any biology. Okay, now what? Can I do it?
You could use, then you could use mathematical methods, singular value decomposition.
Okay, never take mathematical methods to be a beautiful black box that one doesn't have to understand and something would come out.
No.
I'm asking you to look into the mathematical methods. Okay, so why am I doing this? I'm actually setting up a false choice. Can you do it or can you not do it? There are many layers at which you cannot do it, right? So, okay, let's actually think back a little bit in sort of invoke like a Gedanken experiment, some very complicated genetic network. Imagine there is a genetic network that depends on like thousands and thousands of genes, okay, all interacting together to control the expression of this particular poor gene here, right? It's a rather non-sparse genetic network, right? Then what would happen? Then maybe several thousands of these SNPs can affect this gene expression, right? Now, is there any chance by just looking at 100 strains, I can get any statistical significance at all? Okay? Why not? I'll rarely ever see okay, that a constellation, even if it is true that 5,000 of them together conspired to make a gene expression high, that 5,000 combination, okay, would be seen only once and gone, right? So if there is a very, very, please, come back. Okay, I need, it came back, thank you. So there is aspect one, which is that if I have a very complex system, a system that depends on a lot of things, you can argue why that is not the case, and maybe your prior knowledge is some bit of it, but these days there are sort of all the fancy journals are full of big fuzzballs of things interacting with each other, so who knows, maybe things depend on everything, right, in a very non-trivial way, right, that every particular combination has its own genetic expression pattern, then we are dead, we cannot go anywhere, okay? Okay, so very complicated networks, we have trouble. Okay. Am I pressing something by chance? Yes. Okay. Someone is really bored. Okay. Okay. Now we kind of maybe let us take the other extreme, right? That okay, it's never like a combination of 1,000 mutations together that decides whether some gene expression is up or down, then we are dead. So let's be optimistic and say that's not the case. Okay, maybe most of the time one SNP has an effect, and in the very complicated cases, one SNP has one effect, the second SNP has some effect, and you kind of keep on adding them. Okay, so that's the simplest model. Maybe people will do that for corn heights and your heights, saying that there are many different loci have contributions, and things are just sums of contributions for everything. So this is sort of the simplest model, right? Kind of the linear model, and the other end was like a very nonlinear model. Everybody interacts together to do something. Okay, so in that linear model, are we okay? Okay, imagine every gene, let's think of one gene, let's not take the 6,000 gene distraction. A single gene its expression is affected possibly by every SNP, but I tell you something very restricting, okay, that the effect on the gene is additive from all of these SNPs, so, right? So all I need to know is influences of SNP number 1 on my gene, SNP number 2 on second gene, okay, it's a whole bunch of numbers, okay. I need about 100,000 numbers to figure out, and once I've done, I've gone home, right? Okay, can I do that even with this train number? Okay, now let us count variables, okay. Again, I said every gene its own thing. I have 100 stains, meaning I had this little cell, I grew it overnight into this test tube, I've run out of test tube after hundreds of them, okay. I have sent them for sequencing and finished off my grant money, okay. So in each of those cases, I have gotten some data, I'm very excited. That means for each gene, I have gotten only 100 pieces of data, okay. And how many things are you asking me to determine out of it? You're asking me to determine about 100,000, okay, of numbers of influences. Can I do that? Okay. So in theory, so anyway, you kind of gave me the answer, or I sort of wanted it very badly, to say that the model is generally very underdetermined. If the model complexity is very high in the sense that many genes synergistically produce something, then we are completely out of luck. But even if it was not very synergistic, it was sort of little additive effects from many genes, if everybody had some effect and that had to be added, it is still pretty hard to determine. So the only circumstances in which we have any hope of doing something, that's the case when each gene is affected by a small number of SNPs, okay? Perhaps less than 100 other things. If there is an effect that is more complicated than that, spread among this, we are not going to determine it. Okay? So by nature, this kind of analysis is possible only if in some basis or the other, okay, your biological effects are somewhat sparse. Okay? So that's something that people go with. Now, how does it show up in the behavior of regular population geneticists? Okay, the way, let me see whether it's the next one. Okay, maybe I'll jump up. If you can see that, sorry, this is terrible to look at. But generally, if you ever read a population genetics paper, there will be plots like this. Somewhere you go move along the chromosome, and you kind of try to figure out whether this location, if you just have one SNP having some influence on this thing, you make that as a hypothesis, you calculate some p-values, what's the likelihood that by chance, okay, there is such an imbalance, like, okay, you have, for example, that this particular SNP was correlated with high production or low production. You have some log probability, LOD score, and that jumps up and down along the gene, and you look at these peaks and say, aha, this locus has something, and that locus has something. So you are kind of arguing for most places not having any effect at all, and a few little loci having an effect, and you are trying to pick things up that way. Okay, that is kind of built in our at least a population geneticist brain. So let me backtrack here a little bit. Okay, so in some sense, to learn something, we make assumptions about, okay. Okay, let me keep it somewhere else and see whether it randomly doesn't do anything. Okay, so
excuse me.
Yeah.
One more thought.
Yeah.
If the SNP occurs, would it depend on the periodicity and the breaks from it, where the singularities are in the genome, and how they influence what you know about the transcription factors, their periodicities, because finally the shapes matter. So where the SNP occurs, the folds and other things. So where the SNP occurs and where it plays a part, I mean, which periodicities it plays a part. So some kind of Fourier transform, and that will anyway help in the reduction.
Okay, so I think, okay, so find SNPs in coding region and then try to say whether, right.
No, not just that. I mean, I am saying depending on the periodicities, I mean, the
periodicity
and where the breaks occur. I mean, you kind of from
breaks of what?
See, you know the pattern, you know what the protein or the.
What if the SNP is outside the protein? Much of our genome.
No, not that way. I mean, saying the folds. I mean, the periodicities, they will matter.
But you are really thinking about SNPs on the coding part of the gene.
No, I am not.
So what is fold of something that is out of
see,
the?
Because if this folds, this will also fold.
You are thinking of folding of DNA or RNA or?
I mean, it's related, right? I mean, I'm thinking.
Right, but many SNPs would not, first of all, it's double-stranded DNA, so it may not even fold in any other way.
I'm saying the periodicities may matter. The sequence periodicities, I'm assuming it translates in some way to shape.
Okay, so, okay, look, let us do it offline. Why? Because many people thought that they will do this analysis and it will be immediately obvious to them what these SNPs are. Okay, like it changed the transcription factor, it changed something or the other. The first experience is or it changed the regulatory site. Okay, the first thing is that you find those significant somethings. You even look for correlated SNPs in this block, right? That has not been decorrelated, and you don't always make sense of why those SNPs cause anything at all. Okay, so. Okay, let's have the discussion. Can you immediately see the molecular biology of why a significant SNP does it, right? A fraction of SNPs do that. You can argue why they're doing something, but a large number of SNPs, first of all, as you said, SNPs are correlated. So usually there's a block where there are correlated SNPs and you don't know who the guilty party is. Okay, you often know there's a collection of SNPs together are correlated with something, right? So it's a bit tricky. Okay, but I'm just saying that can you even spot those? Okay, and then trying to explain that, you are saying that can I use molecular biology to sort of guess what their function is or who they are?
No, I am saying you could look at the shapes basically.
Shapes of what?
The DNA, the transcribed protein, whatever. I mean.
Yeah, but some cases they are in between genes and there is no obvious anything associated with it.
That may also play a part, right?
You know, I'm sure it plays a part, but I have no direct way of thinking about how a protein or something changes. But we can talk about it afterwards, okay? Okay. So what's the idea? Okay, so I think what I'm trying to say is that quite often in biology, I don't want to be too general because then it's very vague. But here is a concrete example where I have genetic variation and I have a phenotype like gene expression, or it could be a phenotype like it could even be a qualitative phenotype. It could be a person and you say whether somebody has this disease or not. Okay, I ask for some predicting model, and as I argued, we can have many different kinds of predicting models, right? So there is sort of a very general, vague, abstract way of talking about it, that here is some data, okay? Here is this M represents model, and it could be all kinds of things, whether they're very nonlinear interactions, very linear interactions, whatnot, right? What kind of functions are used to predict anything. And then normally people don't like using X for parameters, okay? I'll keep on using X because I like it somehow. But anyway, these are parameters in the model. So different models could have different number of parameters, okay? The parameters could be of different kinds. Some could be real, some could be, maybe integers, some could be something else, right? So there is a whole diversity of things. And often when we are trying to solve a problem in biology, or at least trying to give statistical description of something, I have to choose among those, right? And how do you compare and choose among this plethora of possible model structures, and then for each of them maybe try to find the best parameter or very likely parameters, is a vast subject in itself. And I am actually not going to talk about the bigger subject. It will be sort of waste of time for me trying to delve into everything. But we are even trying to look at something more concrete, and I already gave you an example. It's kind of a case where the data is given by, say, a linear combination of some effects, okay? But even then, as you saw, even if the model is so simple, you have an issue of choosing a subset of variables to be relevant. So go back to the previous example. Even in the simplest case where SNPs have just additive effects on a phenotype, okay, a quantitative phenotype in that case, like gene expression, right? I cannot let all 100,000 of them be active, right? I cannot determine all the 100,000 coefficients. I'm going to say that most of them don't have a big effect, and only a few have big effects, right? So what I have, so out to the math problem, I had genotype bit strings of zeros and ones, okay? Phenotypes were some bunch of numbers. I'm thinking now one gene even, okay? So numbers, right, for each strain, okay? And what I have now is also a choice to be made, another bunch of zeros and ones telling you whether a certain variable is relevant or not, okay? And that is a particular corner of model selection called variable selection. So gamma was supposed to be the bunch of ones and zeros that tells me whether some variable is being considered or not. It is of the same size as these X variables, right? The X's are, in that particular example, influences of some gene on some variation on a gene, okay? And is it to be taken into the system or not, okay? So to go back to this concrete example, right? And now what I want to say is that I have some observations, yi, let's say, was gene expression. I write it as sort of an expansion in terms of some variables, okay? So if the single gene model would stop here, okay? GIA basically says in the genotype, okay? So this is the gene expression for a particular gene in the ith strain, okay? Or ith individual. Then in that gene I, you ask, this guy says 1 or 0 for whether the eighth SNP is there or not. So if it is like the majority, GIA would be 0, okay? If it is the minority allele that you see there, it will be 1. XA is the influence of that SNP on the gene expression, right? So I'm trying to estimate this X's, right? And I would have stopped there and, okay. Okay. It has moved to the previous page, okay. Right? But I can expand it to do something. Actually, what motivated me to look at is things like epistasis, where there is non-trivial effect from having two SNPs together that's not explained by kind of additive effect of individual SNPs. But you can kind of continue to write it this way by saying this is the indicator whether the 8th variation is in the ith individual and the bth SNP is in the ith individual, right? So this would be just the indicator that the combination exists, and XAB would be the influence of that on the gene, right? If I had n SNPs, there are n possible influence variables, and here it gets to be n square, and 100,000 square is a nasty number, and if I go further, it'll get even bigger. So if I have phenomena, much as I originally said, the 5,000 gene thingy is either not possible or we don't want to even think about it because we can't do anything about it, right? But in many processes, you will see, just as we learned about genetic networks, we expect 2 or 3 genes to interact together to produce some non-trivial effect. So detecting those are becoming harder and harder because we have very few individuals, okay? And many, many possible potential variables contributing to this, right? So that's our trouble. Now, before I completely forget biology and move on, I gave the example of population genetics and quantitative traits because that's actually staring at our face in many ways. But it's not just restricted to this. So I'm sorry I picked this picture because there are too many things in there. So from far away, I don't know whether you can even focus on this part. But I'll tell you a little bit what this is. This is actually another example of a similar problem. There is a whole industry predicting whether a certain protein would interact with a certain sequence, or even more carefully, if I have a certain amino acid combination there and a certain DNA sequence combination, if I dock one on the other, what would be the free energy of protein-DNA interaction? So if this free energy is very low, then it's a good binder, if not, right? And people have actually gotten serious enough to gather some data on it. So you take some proteins, you mutate some, you do some SELEX-like experiments to fish out some sequences that bind to it. You can do careful KD measurements. So you know free energy for a library of DNA sequences and protein sequences, okay, to figure out what this, what the free energy of interaction is, right? Now, out of that, can you learn the rule of how these things interact, right? So there are 2 kinds of ways of going about it. There is the physics to death way, which is I really have potentials from, I don't know, realistic this simulation or that simulation. I turn them on. I like amber. I like charm. I like whatever. I start doing humongous calculations and try to predict the numbers. Those things go somewhere. Given our accuracy with structures or something, and given the limited accuracy they have, that they barely get anything is impressive, okay? But mostly those are not the cutting edge. You don't run to your chemical physics friend to predict those affinities, okay? They usually catch up, okay? So that's one way. The other way is completely statistical, right? Which is to say, well, let us make some statistical correlation models and do some data analysis and see whether I can say something about this affinity, which is what many bioinformaticians do. So here is this very oddball effort where people say, okay, I have these 2 molecule structures on top of each other. There are a whole bunch of carbons and nitrogens. They have some interaction potential that is position dependent, okay? Only thing I know about it is that if this interaction potential, you go few distance units away, the interaction is nearly 0. So imagine you say there is a certain distance and there are some bins for different distances, and each of these bins have a different free energy of interaction, okay? And you want to learn it out of that data, right? You have the same kind of trouble now. This potential VR between molecule I and molecule J, even if I make very crude bins, have many possible numbers in there. Okay? And then there are many pairs. So many pairs multiplied by many distances produces a humongous number, and we feel like giving up. Okay? And I'm going to learn it from data. How? Right? Okay? Now, if you think again of the previous example and think about your intuition about these interactions, you could say though, yes, it is many, many, many numbers, but many, many, many of those numbers are most probably zero, nearly zero. Okay? So these are hydrogen bonds and this and that, so most of the time very far away molecules really would not have an interaction. And even close by guys, if I move them out a little bit, most probably wouldn't have much of an interaction. So you can write this interaction in terms of additive models, in terms of this funny V of R of T's, right? And assume that only some of them are non-zero, okay? And you can turn, you are in the same ballpark as what we are trying to do for genetics. We are trying to say, I have these predictions to be made out of many possible variables, but only few of them contribute, okay? So I hopefully made an argument for looking at this kind of underdetermined problems, okay? And rest of my discussion would be about these underdetermined problems, about their successes and limitations and whatnot, okay? Okay, so, well, maybe before scary math, okay. Any questions so far? Okay, yeah. Yeah. Yeah.
So, supposing you were told that out of these 100,000 SNPs, that is affecting this particular gene I.
Right.
Yeah. So, if you knew that, if you were told that 100,000 out of 100,000 SNPs, only of the order of 10 were actually affecting gene I, but you didn't know which 10 they were.
Right.
Then, would you be able to find, I mean, the question would be that if you know that, or if you make that assumption, then can you find which 10 and what their effect is from 100?
You shouldn't be stealing my thunder. Okay, yes. Okay, so that's a good question, right? Can I? The answer more or less is yes, and we will be discussing whys and hows. Right, okay, let me actually go ahead and tell you something, right? Because I have already discussed this problem to some extent. So there are many ways, so Sanjay says that there are this many things, but I tell you 10 out of all of these things, okay, affect you, okay? How does he know that? I don't know, but 10 is maybe his favorite number, okay? How can I do that? So the traditional way with log odds is sort of saying do the following. Look at each SNP and see whether you are significant, right? So there are peaks. So I can always count down from the highest peak to the 10 peaks, right? And in some cases, this would be a fine enough way of discovering those 10. The 10 most significant peaks in this LOD, okay, plot across the chromosome, right? Now, what you may be asking though, okay, by looking at the top most significant one first, which is practically a one SNP model, okay, is this a good idea to sort of say, I try 10 one SNP models, or I try actually 100 million one SNP models, pick the top 10 one, or I allow for the possibility that weak effects from 10 of them together make something significant, am I actually going to miss that out? Okay? So if there is nearly equal weak effects from something where together they are significant, there is a chance that that will hide under noise. If you say one guy is significant over everybody, one guy is significant over everybody, right? And that's a subtle difference, right? But that's what we are trying to do. If you tell me 10, what's the best way of recovering those? Okay? So hold on, we'll come back to it. Okay, so let me jump forward and pose this underdeterminedness problem in the following way, okay? Suppose I actually had some vector, okay? This vector is very high dimensional. We saw the genetic thing in the simplest problem is 100,000 dimensional, maybe 100,000 square dimensional, okay, right? But most of its entries are small. Only a small fraction, okay, is non-trivial significant, okay? I multiply this thing by some matrix, okay, that's taking linear combinations, and generate a bunch of numbers. And these bunch of numbers are usually the dimension of this vector is much smaller than the dimension of this vector, okay? I'll have pictures to impress it upon your mind, okay? But this is sort of the typical underdetermined linear system, you can tell me why if I can find infinitely many x, right, which will solve this problem, and then, that is the ambiguity in the system, okay? But if I could actually tell you something more, as we said, that there are only k elements of this which are non-zero. So I'm taking an extreme problem, right? In real life, maybe nothing is zero. Okay, you know what? This is a terrible one. Let me just jump in through this, okay? Maybe this is more helpful, right? Okay, n is the big vector. Think number of SNPs, number of potential biological causes. M is number of measurements. And usually you have this very oblong matrix converting this to that, okay, right? And generally, in linear algebra, you would not be asked to solve for x, given y, because there are too many answers, okay? Right? But the thing I'm telling you in advance that your general linear algebra instructor did not do is that only a few of these n things, so there are these blue bodies, right? Those are the k non-zero entries. Most entries are 0. And k is less than m, okay? So now can I solve this problem? Now, turns out that this is a rather important problem that appears in many places, right? It originally appeared when people were trying to figure out the following thing. Geophysicists were sort of thinking that I'll send waves through so you send sound or some other waves through a region, and depending on whether there's an oil deposit or some mineral deposit, your waves would behave in a certain way. If it is plain ground rock, it behaves in another way. And you can kind of ask sort of this, it's an inverse problem by seeing how the sounds come out. Can I figure something out? And in general, of course, Earth could have been very complicated, and you would not know anything by measuring sound waves somewhere else, right? But if it is like a few lumps here and there and not very specific, right, you can reconstruct things. And there are a whole bunch of problems which are in principle undoable, but with certain constraints doable, right? We get told how X-ray crystallography, knowing the intensity, you could not have reconstructed, right, what the molecules were, because you just got the convolution of the Fourier transform, not the Fourier transform. But if you think, okay, Watson and Crick ended up solving the DNA structure, or Linus Pauling ended up solving something. Why so? Because they were not looking at the arbitrary density profile, which could be both positive and negative. They were looking at very lumpy, okay, few balls. So, I mean, in some sense, if you think about it, the atomic model, okay, a bunch of atoms, Fourier transform of that mod square, you have some hope of actually guessing what it is. And with a few perturbations, you can definitely guess something, right? And in many cases, these constraint problems are much more soluble, okay, than superficially it looks like. Okay, so the recent bang has been looking at applications in signal and image processing. I think this picture goes by the name of, or the woman goes by the name of Lena or something, that shows up almost in every third paper on compressed sensing. Someone takes her pictures and doodles on it. And then somehow you have to know that in wavelet space things are sparse, which possibly had to do with that her skin is really not like that. Okay, and there are some smoothness, which in some Fourier, local Fourier-like basis means only few components are active, right? By using that, you can actually do a pretty good job of reconstructing things, right? So actually in some space, right, things are sparse. And if you know that, you can do a reconstructing. Now, there has been a flurry of activity in computational, statistical, and sort of engineering circuits. But in that process, many of them have noticed these sharp transitions and have adopted things like replica trick and other things that physicists used to use for disordered system. So now there is kind of a growing community of physicists who are trying to understand nature of success of these algorithms. And there is some real opportunity of figuring out whether difficult problems in biology, which superficially looks insoluble, is there a corner out in the parameter space where you can actually apply these methods. Okay, so back to this picture. Okay, so here is my time again to sort of see how many of you are asleep. Okay, Right. I think, did I ask you, any of you? No, no. Okay, question is that can I actually, do you think I can solve this problem? Okay, kind of Sanjay's question. I have n variable, n larger than m. I have k of them non-zero, okay, and k is, of course, smaller than m, right? Is there a hope? Can I actually, and I tell you honestly, the y I give you.
If it was h times some x?
Right.
Yes, but if it was h times some x plus any amount of noise, I would say no.
But with noise, you will never reconstruct exactly. So then you are asking about how much error with which you will constructing. So let me give you the noise-free question. Okay. Okay. So, well, tell me something, because when you feel confused or something, what should you do? Take kind of the simplest problem. Okay? Right? Okay, let's keep m and n, right? Let's take k to be 1, right? If I can solve k equals 1, I can't solve anything. Okay? Do you think that can be solved? How do you go about solving? Your exam coming, right? Someone has, you pass or fail based on that. You are willing to do any amount of work for it. Okay? How do you do this?
Hello?
Okay.
Yeah.
Yeah.
You can frame it as an optimization problem where actually you're maximizing the entropy of H or something like that, then the solver.
I have given you H.
Oh,
okay, exam has the H written down. Okay, it's a rather full-looking matrix. Okay, there is nothing crazy about its rank. Its rank is M. M is lower than N, right? Yes, we can do it as some fancy optimization problem, and I'll come to that, right? But I want you to find out. I said k equals 1. Sanjay likes k equals 10. I make it easier. Okay, can you actually find which one is non-zero, and then of course you can find which is x. Well, what's sort of the dumbest way of going about it? It's easy to say dumbest, but then, of course, it's hard to find the dumbest even sometimes. Okay.
We can
Yeah.
do the PC.
We're given both y and h, and you're trying to construct x. So y, think again, in my first example, these were gene expressions of a particular gene. These are ones and zeros in the table saying, okay, whether a so this is an yeast strain gene expression, yeast strain gene expression, yeast strain gene expression, yeast strain gene expression. This is the first SNP there, not there. First SNP there, not there. Second SNP there, not there. So it's like SNP existence, ones and zeros, a table made of that. These are the influence of the first SNP on gene, right? On each of these strains, which is strain independent, right? If the SNP has an effect on that gene, it has it in every strain. Okay. You guys are too knowledgeable. Okay. Okay. So the dumbest thing to do would be to just try first coordinate, second coordinate, third coordinate, fourth coordinate. But notice what's happening. Okay. If it is, so if I say this is the non-zero one, okay, and there is no noise. I know everything in life has noise, but one thing that physics teaches us, and some bit of mathematics, is take the ideal example first and think of everything as perturbations of ideal examples. Okay, and something stays. Okay, then what would be picked up? Only the first row would be picked up. Sorry, first column would be picked up. Okay, this column either is proportional to this y vector, so the y vector is pointing out in some 100-dimensional space. Either it's perfectly aligned with that or not. If it is not perfectly aligned, throw it away, right? Move to the second one. If I really guaranteed you somebody is zero, one day click, okay, your column would completely line up with the vector of gene expressions, and you will go home happy. Okay? So if it was k is 1, it was done. Okay? So now you're excited. Now this problem 1.b, okay? K is equal to 2. Okay? What are you going to do? You have tasted blood now, right? Well, the answer is obvious. You're going to tie two combinations, and generally, this is the idea. Okay? If I set, okay, now you can be a little bit more sophisticated and do diagonalization and whatnot. You choose a particular subspace, And that's your hypothesis, that those are the non-zero ones. Okay? Suddenly, it has gotten a very, very, very reduced version. This H has now been shrunken to some H reduced. Okay? There are only few X's active. I now have many observations. Okay? I have more observations than variables for each of those choices. Right? And in general, it's overdetermined. Right? Just like if I had a vector and I was trying to align it that way, I'm not going to do it unless it's perfectly aligned. For two vectors, they can actually span a plane. If your original vector was out of it, you are dead. So look at various 2's, and someday you will fall on it. And there, if there is noise, then of course at some point there is confusion zone. Okay? And you would have trouble, but if it was zero noise, we'll be able to do that. Okay? So my lecture is over, right? We have found the way. Okay? Just randomly try, okay, various combinations of m possible variable that could be non-zero. And in general, it is going to be an overdetermined equation. You are not going to be able to find, okay, a good solution. You will not find a sparse solution, actually, right? And, oh, sorry, I'm talking the wrong way. Okay, so I could go by k, and I could declare this to be 2, 0 or not. Okay, I could go the other way too. I could pick m of these n things. Okay, I can construct a reduced problem, and I can invert to solve, and I'll find it's not k sparse. Okay, so there are, oh, sorry. So I was saying k equals 1 and look through everything, k equals 2, look through everything, right? That's one way of going about. Okay, actually, let me stick to that line of argument. Why is this not the solution of the problem? Okay, Sanjay wants to do 10 sparse. I'll go up to 10 and find a solution. Okay, why would it be problematic ever? Why is this a problem even? Huh? No. I can fit only one solution. But look, if I do, okay, if k is something, I have to look at k and choose k possible solutions, right? And for n equal to 100,000, choose 10 is already a little bit high end. And if it is more than 10, I'm getting into trouble. So my trouble is that if I really push it to k's that are beyond 1, 2, 3, okay, I have to look at a very large number of possibilities. So my brute force, look at all possibilities, okay, and a way of doing things is not going to work for me. Okay? And this was another way of arguing the same thing. This is a little bit more straightforward. Here you don't say, I take the k that are non-zero. You say, k is less than m. I'll pick an m number of things, set everything else to 0. Now I have a square matrix problem. I have m variables I've chosen to be determined. I have m observations. The reduced problem, the H reduced to that space is a square matrix, so I can solve it, right? But then I find that this X practically is non-zero for every component. But I said only k of them less than m was non-zero, so it will be inconsistent. So I can look that way too, and I'll soon figure out that only one of those choices is consistent. Okay? But the trouble with all of these problems is that it looks for a huge number of combinatorial possibilities. Okay. So now we have solved the problem and we have not solved the problem. The problem in principle has an answer for most H's and most Y's. Okay? But getting to it efficiently is the problem. Okay? Okay. So everyone is with me so far? Okay? Okay. So here comes then the So this is maybe, even this guy doesn't like my slide. Wake up, wake up, wake up. Or maybe don't wake up. You can see some monitor still has. Okay. I should have done that with animation. Okay. So, okay, let me try to tell you the idea. Okay, and then I'll go through the steps of it. I realize now it's too busy a slide. Okay, so we kind of come to this conclusion that that looking at all kinds of choices of options in principle would solve it, but is a difficult thing. Okay? So now what can we do? Okay. So here comes a slightly oddball idea. Okay? So we have decided that if I give you the problem, choose a k-sparse solution of y equal to something, right? Y equal to hx when there is such a solution. I can try to set it up as an optimization problem that you suggested, but solving that optimization problem. Okay? So for example, I could do the following. I could say, here is my loss function, y minus hx whole square. That has to be as small as possible. Well, we know that can be 0 over many, many, many things because it's underdetermined, right? I give you another function added to it, which is kind of something that counts the number of non-zero components, okay? And put it with minus sign with a coefficient in front. Then, of course, I would choose the sparsest solution among all these guys, and that will be my victory lap, okay? The trouble is that you can begin to see the geometry of this thing, okay? What's happening is that this, so maybe I should hop forward and try to do an easy example, okay? The easy example would be one measurement, two variables, right? Okay? And I have then a line on which I can solve all of this, okay? But then the sparser solutions are this and this, right? And they are kind of few and far between, right? So if I'm with a dumb model moving around here, I don't know that I'm going to find a sparse solution close by, right? It looks very easy here, but in practice, it's exactly like the golf holes in a flat landscape situation. So this is not a good way of doing the optimization. So what is a good way? Well, the good way, at least one of the good ways, there are many, okay, seems to be to replace this problem by another problem, okay? And the one that created quite a bit of stir is the following, okay? So there is something called lasso, okay? And I will perhaps in the third lecture pick up something that was the prehistory of lasso, okay, about how if you have an underdetermined system, how do you try to control all these parts all the things that we cannot determine, right? But the idea here is that I have this minimization as my constraint that hx has to be as close to y, but I add a regularization term. And the regularization term I add is basically take with some coefficient, just take modulus of each of these vectors, okay? And just add them up. So I have modified my original optimization problem with many, which has many solutions. Many things would have this to be 0, and added something else to it. And I want to find the minimum of this new problem, okay? So what are some of the good things about this function, okay? One thing you notice is that, okay, this function is always convex, right? Because the quadratic part of this is actually x transpose h dagger hx, and h dagger h is at least non-negative definite, okay? So it should not have like some directions where it's in a minima and another direction where it's a maximum. But if you think about it, this L1 norm of x1, which is basically just absolute value of x in each of these variables, right? Those are convex functions too, okay? So all I have is this sum of two convex functions. So in theory, okay, this is something that can be solved efficiently, okay? And there are many garden variety, there are many algorithms that are specifically tailored for solving such problems. But if you are lazy, you can play the following trick. And I think maybe I'm going to, okay, how many of you are familiar with convex optimization in some form or the other? Okay. Okay, how do you optimize over a function like this? What would your idea be? That norm 1 of x looks like a nasty guy, right? If you try to differentiate this, life gets difficult. Okay? So it will be nice to be able to avoid such a thing, right? Meaning actually, you can never get anything for free. But actually, there is a trick to reduce it to another class of problems called quadratic programming. Okay? So this is where I feel like having a blackboard, but I'm told that this thing, huh? Finally, huh? Actually, yeah, it is already on the monitor, and most probably the screen would be seen. Should I press this?
I can turn off the screen. Yeah, I'll do that.
Okay. Hopefully we can. Okay, we'll be fine. Yeah, sorry. Yeah.
Is there any intuitive explanation as to why choosing the L1 norm and not the simpler L2 norm, which would be convex as well and would give less trouble?
In fact, maybe the picture later would. Okay, so I think I got another question which is more basic than how to solve this problem. Who cares about how to solve this problem? Is why not add something like an L2 regularization? And it's a time-honored thing to do. It's called ridge regression, right? And it's been done before, right? So think about what we are trying to do. And let's stick to our simplest example. I have one observation and two variables. Okay? So I have, this is x1, this is x2, and I have my only observation to be some linear combination. Okay? And once I specify that, this is most probably dry. Yes, it is. Okay, so I would not use colors, but all over on this line, I have the same solutions, right? I'm looking for sparse solutions. I want to do something which will pick this or that, okay? Now, so if I have this now, y1 minus a x1 minus bx2 whole square, I'm going to put a half or something, that's 0 all over this line, okay? Now, I'm going to add a little regularizer to break the degeneracy and pick a certain point. So imagine I put a teeny lambda, okay? And just to sort of humor you, I do this, which is actually the first thing many people did, and it has its own value, and it's actually a good thing to do in many cases, okay? What's the contour of constant values of this function? Well, they are circles, right? Okay? Which point do you think, if lambda is very small, so that you more or less would stick on this line, okay? And you will just pick one of them. What would you pick? Which point would you pick? Well, you'll pick some point kind of in the middle, right? So it is a perfectly genuine way of trying to break the degeneracy between all the solution. And again, I'll talk later about its virtues and vices. But imagine I was told things are sparse. Okay, and several of things had some influence, right? The ridge regression has kind of a tendency to choose the kind of democratic average of them. So if several of these sparse solutions are competing with each other, it will choose something in the middle, like this, okay? Which by itself is not always a bad thing. But and in this case, perhaps there is some reason for doing that. But imagine this thing was really, okay, very skewed. You had a reason to choose actually this solution, and I'll try to make it more concrete, right? It will actually not give you a sparse solution. Now you can say, I'll do ridge regression, maybe try to drop the smaller ones, and is it very close approximation? That's a fair thing, right? Okay? Okay, anyway. So rough answer, ridge regression doesn't do, okay, sparse solutions. It usually naturally gives this kind of full solution. It does something interesting in a rotated basis, and we'll talk about that later, okay? Okay, so the second thing was about optimizing this function. Okay, so so many of you have heard about linear programming, Have you? Right? So linear programming can often be formulated this way. I have a bunch of linear constraints on x, ax equal to b, and perhaps every component of that satisfies some inequality. Okay, is greater than or equal to 0. Other kinds of linear, well, this is an inequality, you can always introduce extra variables and formulate it this way. Okay, and subject to that, I have some linear function c dot x, and I try to find the maximum or minimum of this function, right? And what happens is that because ax equal to b gets you some kind of affine space, and inequalities very much allows you to have this convexity condition that if there is x1, x2, a convex combination of that is in this field. Usually these things, say again in 2 variables, would have some region like that. The constraints would often be satisfied in a convex region. And then your linear hyperplane perhaps would find a corner to this solution, right? So that is linear programming. Okay? Now, if it is a convex region, linear functions are special kind of convex functions, right? But, if I had a function that is Cx plus x transpose dx, where d is, say, has no negative eigenvalue, right? Okay, then this thing is a convex function too, right? And so think of now a paraboloid coming out this way. I could have situations where the minimum of the paraboloid falls inside the convex region. Sometimes the paraboloid minima falls here, but there is a slice in this direction. Now I'm waving my hand too much. And you will find that inside the convex region, this is the best solution, right? Sometimes the minimum of this function is out there. It's way outside, right? And you will find that the constraint optimization problem would pick some solution like that. So it's a little bit more non-trivial than just taking the derivative and setting it to 0 and solving, right? Because of the inequalities, right? But once you have everything set up, what happens is that you end up getting, you either get a solution where all variables can be varied or some are restricted. And even if this function's derivative is non-zero, if the derivative is perpendicular to that boundary, okay, that would be your constraint optimization solution. And there are whole packages to solve that. It's been an old subject, okay? So there is both linear programming and quadratic programming available to you as kind of well-developed methods to solve things, right? Now, the fun part is that, actually this problem where I have, which is a nice quadratic problem, okay, quadratic function and linear function combined, right, plus this guy, right, right, this could be converted into a quadratic programming problem. And I think I'm showing the page by which you can do that, but I'll sort of try to explain again with 2 variables how things work, okay, and then you can understand the general thing, or rather with 1 variable, right? Imagine I had to deal with an optimization problem where I had this norm of x1 added to some nice quadratic, nice smooth function, which is quadratic and linear, right? But this guy is my problem guy. I don't like him, okay? Now, you can deal with this optimization problem exactly in this general inequality formalism by this way. Write x1 as x1 plus minus x1 minus, okay? So I express this guy as a difference of 2 variables, right? Now, that can be done in many ways, right? I also say that x1 plus is greater than or equal to 0. x1 minus is also greater than or equal to 0. Okay? So, okay, big deal, right? There are still many ways of doing it. But now let me go 1 step further. Let me optimize following function. look, no absolute values needed because I've already constrained them to be positive. Okay? If x1 is fixed, okay, what would be, right, these x1s, what x1 plus and x minus 1 plus choice would minimize this guy? Oh, sorry, right, thank you. Okay, so again, my kind of sleep check. How many of you are sleeping? Okay, I have a number, fixed number. I write it as difference of two positive numbers. Okay, and I want to optimize, okay, the modulus of these two positive numbers. Okay, what's the solution of this problem? Suppose I have 10. I've written 10, huh? x1 plus equal to x1 minus, then the difference is 0. So suppose x1 is 10. I have x plus, x minus, and 10 plus x minus. And I have added their modulus, or added them. Both are positive, right? When would it be the least? Who is 0? It's, okay, it would be when one of them is 0. Who should be 0? You guys have decided x1 minus is always going to be 0. Then I should have never introduced it. When should x1 minus not be 0? Very good. Okay, so if x is positive, then the plus guy should take everything, and the other one should be 0, right? And if it is negative, x minus should take everything, right? And any other arrangement produces greater x plus plus x minus, right? So here is a simple cheat trick, okay, to not have to write absolute value. Of course, the absolute value is hiding in these inequalities, but that's friendlier, okay, to the quadratic form programming or linear programming formalism. So what would happen is that I would actually replace this problem with this problem where I write this as the L1 norm of these two things, but since these are all non-negative, they are just simply this, okay? So I write a function that now looks nice and differentiable, okay? And all the technology of quadratic programming apply to it. I have, the cost I paid is I've doubled the number of variables. But hey, maybe that's not my headache, okay? And then I have these inequalities. All of these are non-negative, right? And that's all, right? So now, and my original, yeah, so I'm done. Okay, so what did I try to do? Okay, there is not, okay, I'll bravely try. Or maybe I can give the talk on the small. If not, we can, yeah. Okay, now stop. Sorry, Mukund. Okay, yeah. Well, I can wave my hands. Okay, great. Thank you, thank you, thank you. Okay, so fine. Oh, why is it going in the? Just 1 second. Yeah, I think that's where I was. Okay, so I think I have explained the trick, okay, that allows you to forget about the norm and reduce it to a quadratic programming problem, and then you can use something to optimize this. But this method of adding the lambda times this thing, it goes by a name of lasso. It sort of was suggested in a bunch of papers by Tipshirani around mid-1990s. The idea was the following, that it's kind of relevant to the problems we are trying to do. Well, if I have like if I have a problem where, in reality, nothing is 0, but some effects are strong and some effects are weak, right? Suppose everything was weak. Something was absolutely, everything was 0. And I did actually what Mukund said, which is always true in nature. You really never get h times x. You get h times x plus some noise, right? And I tried to solve it by some linear regression method. What ends up happening is that I end up fitting noise, right? I often get big coefficients that are related to the matrix structure and the noise structure, okay, right? So the risk, okay, of allowing for many variables, and this is something perhaps we'll talk a little bit more, having a very complex model with many variables, is that if I have limited amount of data, I'm often fitting noise. At least the variables that were small, okay, would be very different from experiment to experiment, okay? So if those variables are there, on the average, I'll still do a good job. My bias would be small, but my variance of these terms relatively would be quite big, okay? So what can you do about it? So one thing to do about it is that scale down every variable a little bit, okay, and especially scale down heavily the ones that are guilty, right, and sort of ridge regression. The L2 norm does it to some extent, and generally this goes by the name of shrinkage, okay. But what Tibshinani pointed out, that if he changes the regularization from the quadratic to the linear one, it also sets certain things to 0, okay. And how that happens is practically my same picture, okay. Let me go there. You see that the constant norm 1 surfaces are these diamonds, right? So if I minimize the diamonds instead of those circles, right, I tend to pick these corners, okay, right? So I actually can do shrinkage and selection, meaning setting some variable 0 at the same time. It's a good thing to do or bad thing to do is another question, okay? So anyway, there is, okay, so I'm going back and forth, but let me talk about the thing that I want to end this lecture with, okay, for today, is a very special case. And the special case is when lambda is very small, which we're kind of arguing about, okay? And what you're trying to do when the lambda going to 0 limit is that you say this is not going to be violated. I'm going to find x so that y is going to be hx. But in that subspace, let me minimize this function, okay? And as you can guess that this, okay, this is what is happening there. This is how I'll pick a solution. But this is even simpler, because what you are optimizing is norm of x sum, and after that difference trick, it's just a linear programming problem, okay? I have a bunch of constraints, bunch of inequalities for each variables, and I have this function to minimize, okay? So algorithmically, this is great, okay? And what somewhere around mid 2000s, a whole bunch of work from Candes, Tao, Donahoe, Tanner, etc., started showing is that in some cases, like that lady with scribble faces, right, you try to even reconstruct somewhat noisy data and you do pretty good job, okay? So there is some observations growing that the following thing happens. They like to call it this way. L0 was the norm which was kind of counting how sparse your matrix is. And I said if I add that as an additional thing in the optimization function, it's very non-convex and hard to do that. If you replace that by another optimization problem where there is this L1 norm added, and it's a convex problem, you can solve it, under some circumstances, you seem to reconstruct the original sparse solution perfectly, okay? And this is a bit of a mystery, but I think the mystery amount has gone down, and we will talk about it tomorrow. But let me see. So let me actually try to set up how we want to think about it. So there are two key variables here, okay? One variable has to do with how many measurements I have, okay, per possible variable. So, so this is m over n. Okay, I should cancel off all these happies and something. Okay, for solving the problem, what does your intuition say? Is m over n large good or m over n small good for reconstructing? You are really sleepy. Okay, is more data good or bad? Usually good, right? Okay, so if you have more observations, it should be somewhat better. So high end of m over n should be good. Okay. Now, we say we kind of depend on the fact that these are sparse. Okay? K is the number of non-zero things. The rho is the other ratio, K over N. Is high K good or low K good? Low K has to be good, right? K equals 1 was our superstar. Okay? So, sparser the better. Okay? That means in this phase diagram, if I could already say that in a thing, this side is good and that side is good, right? Where is the region where I can, if I had infinite amount of time and computer time to trying out every combination, the problem is soluble in principle? That's the regime where K is less than M. If K is less than M, then for most problems, the brute force search solves the problem. That means down below, everything is in principle soluble, right? Now, it turns out, if I look at this and I try this trick, I say my old problem has been forgotten, okay? I replace it by this convex optimization problem, okay? Which finds some solution. You can ask whether this is the right solution or not. Is there a region where it is the right solution? And it turns out that somewhere out there, this starts becoming true, okay? Now, it could be that I have another idea, right? And that would have another boundary where it is true. But I draw this boundary in principle. There need not have been a boundary. It could be just like a crossover, okay? It's bad here, doesn't do a very good job, lots of error, right? You go there, things get a little bit better, right? Could be very smooth. Now, the shock of shock is that actually it's a very sharp transitions. Now, I hate that picture. This is from a rather famous picture of Donovan, paper by Donovan and Tanner. Just to make our life difficult, they are plotting it another way. Okay? So, remember, I should have some way of showing both pages. This is sparsity over number of variables, which was horizontal for me. Somehow I like it that way, right? That's fine, right? So the y-axis here was the x-axis before. But who asked them to plot p over n? P is actually n minus m, right? It's the number of, it's the deficit between number of variables and number of observations. So not only have you got to do x, y change, you have to also flip this axis around. And after that mental jugglery, it should be the same picture. But take it from me, since none of you want to do it, okay? The blue here is probability 1 nearly of reconstruction, okay? And the various greens is the rate at which you are making mistakes, right? How often you do that. It's pretty bad out there, right? And near the boundary, it looks like it's a very sharp boundary. And this is some other, even more oddball way of plotting the same graph, okay, right? Somehow I would have liked them to plot really how much was the error per component as a function of any of the scans. For example, for fixed k over n, if I went like, let's say this way, how does it behave? And I never find a good picture for it. So, okay, we tried to calculate something, and this is out of a very, very, very small data set, right? We have only 200 possible variables, 10 non-zero ones, 10, your favorite number, okay? And here, okay, I start with 100 observations. So alpha is the fraction of observations to that, right? So this is a very large lambda equal to 0.1. And so in here, the strength of the L1, if I make this L1 norm term, MSE, okay, that means mean square error. So I have a vector to start with. I have a vector that I recover. I take component by component this difference, square, right? And I average it over the number of things. So this is my average error per component, okay, right? So, okay, one thing is that since lambda x norm x1 has been so good, why not turn off lambda that's very, very large? What would happen then? What if lambda was infinity? Huh? what would be the solution if lambda is infinity? This is really sleepy test. After 1.5 hours, can you even think straight? What would be the solution of the optimization problem if lambda is infinity? Then the quadratic term doesn't matter. Where does norm 1 minimize? At 0, right? So that term is really shrinking and selecting everything out of the game. So if lambda in this problem, everything is kind of order 1, if lambda is even 0.1, okay, you can kind of see there is no feature at all, right? Because it's choosing a really very sparse, almost nearly 0 solution. So the error is practically the same everywhere, okay? But fun begins if you start reducing lambda. I mean, lambda's job was to be infinitesimal and regulate things, really. And you see that the curves go down, down, down very quickly, and they, okay, this is a finite and whatever calculation, they pretty much go towards a limiting distribution. And you can kind of see that in here the error is rather low, right? And the error starts picking up from here, right? Very much like what you expect in a phase transition. And it turns out, this is something I'll maybe talk about depending upon whether you're throwing shoes at me or not, okay? That in physics there is this concept of an order parameter to measure a transition. And in this thing, in a very natural way, mean square error is the order parameter of this transition, okay? So, okay, I'm kind of close to the end. So I think I would, rather than moving over to the next thing, I'll kind of summarize what I've said. You most probably don't need it. That we have to keep model space complexity in mind, right? And unless you're trying to determine something very simple, is this particular locus important or not? Okay, those can be determined out of limited amount of data. If you want to say, I'll figure out the genetic network out of this much data, right? Usually you are taking up too big a problem because the model space is very undefined and very large, okay? And this data, even if it was good quality, may not be enough. So sometimes it is too little data. We need some way of restricting it, so that could be prior knowledge of biology. It could be some other method that automatically tries to choose it, right? Some cases, some form of sparseness is most probably essential to assume, okay? The sparseness may not be in the space of loci, could be in some other space, right? But it's important. There are parts of the kind of phase space of these problems, which has to do with how many variables, how much sparseness, etc., where actually there could be efficient algorithms to solve the problem, although the principal problem in general is a hard problem. And there are phase transitions in sparse reconstruction. People who have been following things about algorithms, this should not be a surprise to them. Even in the whole subject of NP completeness problem, where you know that sort of K satisfiability is a non-trivial problem, you can actually parameterize the problems with some probabilistic structure. And in some K-sat problems are soluble and some others are not, and there are actually very sharp phase transitions between those regions where it is well soluble or not. Some people would go further and say that it is a bit like sort of in many problems we deal in life, okay, there is sort of very little data to learn. For example, you guys all learned some language, right? Now, if I actually set up language as a general grammar and say, okay, these are the rules of the grammar, figure out the rules of the grammar by the first, I don't know, 1 year of your life and stop saying gaga goo goo, okay? You know, there is not enough data for learning an arbitrary grammar, right? So that bit is true. Now, Chomsky would take a jump and sort of say, oh, that's why, okay, there's universal grammar, and you know, our brain is constructed to learn only certain grammar certain way. But meaning at least as a practicability problem, definitely having a very constrained set of possibilities help you learn, right? And this is another example, at least an example of that. Yes?
So, yeah, you said that. So, in the case of language, you can make a good argument that if language was not like this, we'd be in trouble. But here is an evolutionary reason why the genotype might be sparse.
Okay, so that's actually a rather interesting question, and in fact a very concrete question. So the reason we started thinking of this problem is trying to see how at least some of the fly memory genetic networks work. And as you know, there are some mutants that kill fly memory, this famous rutabaga and whatnot, right? Learning becomes very difficult. They don't remember electric shocks, okay? And then, but you can try to then let other genetic mutations happen to compensate for that phenotype. And you kind of, if you take the extremely sparse molecular biologists take, then there should be a gene A and the opponent gene dying. You are the adenyl cyclase, I am the something else, right? Okay, that mutation should kill it. So you'll find individual chunky genes, right? What happens in some of these experiments is that you don't find such chunky genes, okay? And then, but in fact, in artificial evolution experiments, you do see the flies recover. If you create enough genetic variation and select among them, they seem to recover, okay? So the issue is that in short time, like in an artificial evolution experiment, how much complexity can be generated? And that's a more concrete question. Could you generate a 2,000 gene interaction possibility? So I would actually say that you can definitely develop multi-gene combinations coming through, mostly because of recombination and the way things work in sexual development very quickly, right? How many of them together, how many 3-gene interactions are there? That's sparsity. So this 3-gene space is n cube, but how many n cubes are there of this n cube are there is the key question. And you might set up an evolutionary modeling to answer this question, that in a lifetime of a graduate student, can you sort of, what can you generate, right? But it's actually it is most probably the profound question behind this little machinery tweaking, that what space is sparsity in evolution? So I appreciate your question. Okay, I think I'll shut up. Any question? Yeah.
Is there a version of this that applies to, say, for example, GWAS type data?
This, well, yes. So I kept on saying QTL, 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 because QTL is easier to explain. But a lot of GWAS data is basically, so it's categorical, right? That, you have X, you don't have X, right? But it's often done by a logistic model. So you say probability of your having type 2 diabetes. Okay, that's a disaster, but okay, is 1 over the Fermi function, 1 over 1 plus e to the, some linear combination of things. So in some sense, log of probabilities of doing something should be fit this way. That's not quite how it is done, of course, because you have very few numbers. So you actually do it as a logistic regression, okay, likelihood problem. But its mathematical structure is very similar. Okay, so there is a different convex optimization problem, which in some limits would become a quadratic plus whatever with the high population limit, right? So you can phrase basically GWAS data in this framework with a convex problem with this extra regularization that is trying to make the solution sparse. Now, people have tried that. And there's some argument back and forth that one locus at a time or by hand combinations at a time for epistasis, how does it compare with this kind of method of construction? I mean, it's kind of the bleeding edge at this moment. Yeah. But a short answer is that yes, you can adapt it to GWAS data. Yeah. Oh, yeah. Okay, so, right. So between you and you, right, you have sort of trying to give away what I want to say on the third day, okay? So, right, no, I cannot use chalk here, but there is chalk here. Okay, 1 second. I'll try to sneak in somewhere. Okay, so I'll try to use this corner, please, okay, forgive me. Okay, so here is X and Y axis. What you are saying is that, okay, I know my X1 norm is like this, right? And what if the linear, whatever region of ambiguity was like that, right? And, well, I can argue what the L1 norm does. It actually picks something degenerate. But you should ask, as you should by common sense, is L1 norm doing a good thing then? Okay? And if you think about it, this is something that happens when I said my matrix rows are all kind of like random, and two long random vectors are almost orthogonal to each other typically. But if they're correlated, if they're exactly in the same direction, this is what would happen. Two variables would get added. Okay? It will be x1 plus x2 times a vector, right? And even when they're somewhat correlated, right, you get these lines which are sort of parallel, right? So yes, if it is not exactly the same, if it is slightly tilted this way, L1 would pick one of the solutions. But if the data was a little bit different, it will pick a very different solution, right? So you are already sort of putting doubts in people's mind whether in such cases they should trust this L1 norm optimization much as it's promising victory for you. Okay? And in many of those cases, doing what you would suggest, add x squared, so pick the middle, right? They are kind of far away from either solution. But on the other hand, a biologist might say, I don't want to get you a regression function. I just want to know which genes or which genetic loci are maybe important, right? What L1 norm is going to end up doing is pick locus 1 or locus 2, okay? Kind of randomly, okay? Whereas this guy would say both locus are important, I gave them some weight. So if you wanted to make a list of loci that may be important, your list would be fatter, but it will most probably include both possible solutions. And so there is a hybrid between ridge regression with a quadratic thing and the L1 norm lasso-like thing called elastic net. And it's kind of sold on those lines that some cases lasso, especially for correlated features, okay, ends up choosing one over the other somewhat randomly. Meaning a concrete case for GWAS-like data is that if I did not try to break it up, okay, regions with no linkage or very low linkage, right? Close by SNPs are kind of correlated, right? So they are present most of the time the same way, and L1 might choose one SNP over the other, but there is no reason to believe that this SNP is any more important than a SNP like only 100 bases away. Okay, I can't tell really. That's the better answer. So yeah, so there is some case for not taking it too far. Okay.
Okay, let's thank Anirvan one more time.